Hello, this is Nick Cap, and this is the Supplemental Industrial Biotechnological Biotechnical Microbiology. And another name for this would be microbial manufacturing. Another name for this could be applied microbiology. Another name could be your practical microbiology. Basically, all of these things are, again, not necessarily medical related, although kind of everything does relate to our health. These are topics that we cover more thoroughly at Skyline College in a couple of classes that you may be interested. Biotech 400, which is an introduction to biomanufacturing. We have Biotech 170, 171, which are similar to the Biology 100 series, and they do introduce you to biotechnology. Those are being offered next semester. And then there's a Biotechnology 220, which is looking at the business and regulatory aspects of biotechnology. So if you get interested in these kind of things, you can kind of look at it. And I know a lot of you kind of want to go into the medical field, but again, realize that kind of all fields kind of change, and that we're always going to need people to kind of hold people's hands and help them with the medicine. But if you're really interested in the medicine, we need lots and lots of people who are out there who are designing things to help cure people. And I think even more, we're out there helping getting people materials to design things to prevent a lot of these diseases from happening kind of in the first place. And again, there's always vaccines. And so applied microbiology defined. I went to a conference one time, and a guy kind of stood up and, what is biotechnology? And he said, biotechnology is using microorganisms to make money. We want to be a little bit more nuanced than that. And so applied microbiology is basically the use of microorganisms, cells, or cell components to make a product or to provide a service. And so Genentech, which by the Skyline College, is just right next to Skyline College, they produce a number, Herceptin, a number of things that are made by microorganisms that deal with our microbiology that could cure cancer. Okay, and you're looking at curing diseases that before had absolutely no cure. And so microbiology is essential in kind of getting that product to the market. When I look at things, in the past, about 1/5 of manufacturing jobs in the Bay Area are what I would consider applied microbiology related. This includes both governmental agencies and private. Again, a lot more of the money is going to be from the private, but governmental agencies like sewage treatment plants use lots of microbiology. A lot of the regulatory aspects actually use microbiology, and I should have a slide coming in here. San Francisco and Northern California, but basically we're looking at in California, there's over 3,000 what we would call life sciences or biology related companies that are out there. They employ over 96,000 people, and the average pay for these is about 151,000. And so again, being a nurse, can you make this being a nurse? Yeah, you can, but you work pretty hard on doing that. On these kind of jobs, basically they're kind of a little bit more discovery and things like that. So I would say they're a little bit easier. And then also, you're helping thousands of people rather than just kind of one or two kind of into bedside. But what I want to do is, just say that there's lots of microorganisms out there that we have no idea what they are and kind of what they do. These organisms represent an almost limitless, no limit supply of enzymatic reactions. And so in the last couple of days, we've been talking about kind of how enzymes do work for the inside of the cells, but those enzymes can also do work for us as well. And so we think about all the stuff that we do on a day-to-day basis or that we do on an industrial scale, and if we could get microorganisms, if we could get enzymes to do it, we'd be a little bit better off. Okay, so the reason why I would say is that this may reduce the risk and complexities of industrial synthesis. So right now we have to use things like sodium hydroxide and other very toxic chemicals and heat and pressure in order to make things. And again, it wouldn't be great if we used a microorganism to do this. Number one, it would be less expensive, right? We wouldn't have to use these very expensive chemicals. We wouldn't have to get rid of those chemicals after we're done with those. Again, like I said here, the byproducts are usually less toxic. Again, we could actually use microorganisms to clean things up. I don't know if you've ever gone past, there's, right if you go down Sharp Park Boulevard and kind of Sharp Park and El Camino Real, there was an old gas station there. And so you may notice that for a while there, what they did is they just pumped some water and they pumped some microorganisms in there, and the microorganisms kind of ate up all the hydrocarbons that were in that area, and that cost almost nothing. Whereas digging that all out, bringing it to the landfill would have cost, you know, millions of dollars, possibly even for a little area like that. So again, they can even be used in microorganisms. You know, and many times microorganisms are already doing this work, and microorganisms do a lot of work for us anyways. You know, so we look at things like our oxygen supply and stuff like that. And the other great thing about microorganisms are what I call self-replicating. We've talked about this before, and we got kind of this language before. And you know, that's good and a bad thing about microorganisms, because again, diseases can spread because they replicate and they can spread. But the great thing about it is once you get a system set up with microorganisms, it can actually feed on itself. Second thing I want to talk about is this thing we call feedstock chemicals. Feedstock chemicals right now are the chemicals that actually we use, it's called any unprocessed material used to supply a manufacturing process. And so think about all the plastics that we have, the materials for the clothing. All these depend on some of these basic chemicals, and these are great. But right now, these basic chemicals come from our hydrocarbon, basically our oil and things like that. And what's driving our oil is basically using it as fuel for our cars, which is kind of a waste of this materials. And once we burn it up in our cars, it's gone. It's not there to make all the lovely plastics and stuff that we have. And so I just want to say is that a lot of this stuff does originally come from carbon dioxide, and we can use plants and we can use microorganisms in order to make a lot of these feedstock chemicals that we have. We produce lots of microorganisms commercially. Fermentation projects, including beer and wine, are multi-billion-dollar industries that we have a lot. Napa Valley in the Bay Area here is very important, and I would consider that part of our biomanufacturing. We also have some biomass where the physical microbe is actually the things that we want. And so in the Bay Area here, we produce bakers and brewers yeast that people use. There used to be a kind of a Red Star factory when you had on Highway 80, but that's kind of no longer there. We can actually use microorganisms to produce edible proteins. And if you've ever eaten spirulina, that is a type of blue-green algae. Again, these kind of things are expensive to make, and you know they're expensive to make in the Bay Area, right? Just because basically land is so expensive. And in these areas here, you could go out to the Central Valley, you can go out to the deserts of Arizona, and you can make lots of these things, especially like spirulina. You get a little bit of water, you got lots of sunlight in there, boom, you got a lot of product that's in there with very little input in there. We also can make what we call single-cell protein. That is, bacteria can be a source of protein. But trouble is they tend to concentrate toxic compounds. And so back in the 70s when we had lots of petroleum, we were actually using petroleum to make single-cell protein, and it was actually concentrating some of the toxins from in there. Nucleic acids, you know, that's kind of one of those organic molecules that we tend to not think about, right? We always think about carbohydrates, we always think about lipids, we always think about protein, but we never think about nucleic acids. Nucleic acids in large amounts can be toxic. So even though they're everywhere, they're not a lot, right? In a cell, you're going to have the nucleus be 1/10 of the cell, and everything else is 90%. Whereas in bacteria, you tend to see that nucleus is about 50% of the cell, and everything else makes a little bit less. So again, if we eat microorganisms, we always have to worry about there being too much of this nucleotides in there and having a little bit of that toxic effects. Microorganisms can also practice something that we call biotransformation, or what we call bioconversion. And that is when we do our chemical reactions, they can finish our chemical reactions for us. Or what they can do is they can take one compound and they convert it into another. And again, a classic example of biotransformation would be something like converting sugars into ethanol, or converting starch into ethanol, in which we can kind of drive our car. And so it's the same carbon molecule, but we alter it a little bit and use a little bit of the energy, and then we get ethanol from it. The big thing that we have on that is production of steroids from plant precursors. So if you have ever used any steroids, you got a rash and you needed to use a topical steroid, that was discovered, and we manufacture that using microorganisms. What we call, you know, how do we use microorganisms right now? And one of the things is we saw right about the Second World War, we saw the production of fermenters, and so we were able to make these kind of large pressurized tanks. And I think some of this came from the aircraft industry. So we were able to make these aircrafts, these pressurized tanks that fly through the air. And again, if you take that same pressurized tank and you blow air through it, you can grow microorganisms in it. And so fermenters are structures that are designed to optimize the growth and conditions of specific organisms that we want. And in the old days, a fermenter would be just a big tank that you threw a bunch of sugar in there and threw some yeast in there, and you got some alcohol out of it. But nowadays, fermenters have very controlled environments where we control the oxygen, the pH, the medium, the temperature, the nutrients. Foaming can happen in these when you have production of proteins. You have some foams being made. We have two main types of tanks, what we call a stirred tank reactor and a airlift reactor a little bit. This is an example of a stirred tank, and we have a more modern unit of this on here. And so we have a motor that runs an impeller. We have a media in there. We could heat this up. Here's the air going out. Here's the sample port. Here's where we can control all the materials. This is very big. I mean, sorry, it looks kind of very phallic to me, but this is, that's a person right there, and this is a tank. And basically what this is, an airlift tank. And so driving the motor is pretty hard, but what you can do is when you put air, we put pressurized air in the bottom of this, that air will lift up through the tank, and that will actually move the material. So the airlift tanks a little bit. This does get very complex very quick. This is a bioreactor very similar to the ones that they have at Genentech. Again, you go to a brewery, you'll see something a lot like this. They don't need to have all these valves and stuff like that. They don't have to control the oxygen as much like that, but we will see tanks like this. And if you go to your local brewery, which I do, I used to, I don't anymore. And then this is what we call a tank, and so we can impel, we can mix area, we can put sterile air in, and we can feed the microorganisms, and we can pull sterile air out. There will be some materials in here due to metabolism. We'll see some heating in this material. We can use a cooling jacket in order to control the temperature inside of there. These are examples of some airlift reactors. I did show you an airlift reactor that was basically about 4 stories tall. Here are some smaller airlift reactors that we have. The great thing about those airlift reactors, so basically you have an internal sleeve and you have an external sleeve, and so you bubble air up through this internal sleeve, and then this material as it goes up forces the material to go down, and so you have these cycles, and so you have a really good mix inside of there. Nowadays, again, when you have a tank, you have to, when you're done with the tank, you have to clean it up and you have to get it out and stuff like that, and that takes time, that takes money, and you also have to worry about it being clean. And nowadays, a lot of these things are done in clean rooms. If you notice this, this individual right here is wrapped from head to toe in protective gown, and for the most part, it's protecting the product from the germs that are on this particular individuals. And we can actually use single-use fermenters. And so nowadays, what a lot of fermenters are is we have this big plastic bag. Here's the impeller inside of here. Here are all the units for production of oxygen and stuff like that. And so we can grow our, we basically throw a bag into one of these units, and we can grow up our microorganisms in this bag. And then at the end of the day, we can throw the bag out. Ideally, the bag gets recycled, but for right now, they aren't. It's a little bit too hard to recycle them. And so we have basically two ways to kind of grow these microorganisms. And one of these ways that we call is what we call continuous fermentation. And in continuous fermentation, what you're basically doing is you are letting your bacteria grow up and reach that kind of exponential phase. But before they can reach the death phase, right? Remember, they reach the death phase because, or that lag, log, post-exponential growth, they reach that phase because they're running out of nutrients or they're building up some toxins and stuff like that. And what we found out is that you can take out a little bit of the media, add a little bit more, right? Lowers those toxins, raise up those nutrients that you want, the bacteria will continue to grow. And then we can do this type of growth for a very long time, and that's what we call continuous. And so we tend to see a lot of faculties, we want to work on a continuous process rather than batch. And that second one is that batch. So what you mean by a batch is that you would take a tank and you make your beer or whatever in that tank, and then you harvest that tank, package that beer, and then while that's going on, you then clean the tank and get it ready to cycle. And so you go from batch to batch to batch. The trouble is with doing batch is if you don't clean that tank, then you have a lot of problems with it. And so they do have some advantages and disadvantages of these. The other way that I want to talk a little bit more about that we're looking at is that in the past, we really thought that in order to grow things, we had to have pure culture. And what we're finding out we don't necessarily need to have pure culture. And, so things like kombucha and yogurts are made by several microorganisms that are doing the work for you. It's a community of microorganisms. And so as we are getting smarter at using different communities, we are able to make different types of things. The next thing that I want to talk a little bit about is these things, this topic, what I call bioconversions. An example of bioconversion is that of steroids. Prednisone is kind of one of those steroids that we have that was really expensive for a really long time. The chemical synthesis of prednisone takes 37 steps, and if you've ever had chemistry, you lose a lot. So even if you lose 10%, you're ending up with very little. You could start out with 100 kilograms and end up with gram quantities, kilograms, gram quantities of this. The bioconversion requires 11 steps when we use microorganisms, and the great things of microorganisms is that we can control the chirality of it as well, and I won't necessarily get into that. You should kind of look a little bit about how these processes work. Another thing that we can use is immobilized cells, so we localize cells in a matrix. So what's really kind of neat, we can make this a continuous process in that in step number 1, there's 11 steps. We could run this through. It goes through step number 1, we see the first conversion. In step number 2, we see the second conversion. And so basically you can do this, make your substance basically in a pipe. And the pipe is lined with these immobilized cells that go through and do the conversions. Microorganisms are very important in agriculture, and then this is a little bit old technology that I'm talking about here, what we call this ice minus bacteria. And so one of the problems that you have with, here in the Bay Area in our wine industry, it gets a little bit too cold. One of the reasons why we can't grow oranges up here, and oranges grow pretty well up here, but trouble is every once in a while we'll get a frost that will kill the flowers and the buds, and then we don't get our oranges. What's really kind of interesting is where there's a microorganism called Pseudomonas syringae that promotes ice formation. Again, the Pseudomonas wants to eat the fruits, so it wants it to freeze and break and die so it can eat the fruit, and so it actually helps promote the formation of ice. And so what we've done is we've gone through and modified Pseudomonas syringae so that it doesn't have this particular crystal, and so we can squirt those waters on things. And again, this is the Environmental Protection Agency. The EPA is a little bit worried about this, and kind of rightfully so, and so we are looking at it because it is genetically altered. So the Frost Technology Corporation is registered with the EPA as a mixture of naturally occurring bacteria. And so what they've done is they've grabbed some natural bacteria that don't have this, and then what they do is they squirt that on the fields to try and dilute out that microorganisms, the bad microorganisms that's there. And so again, farmers are looking at what microorganisms are in my field, what microorganisms are helping me, what microorganisms are not helping me. How can I alter that content of the microorganisms? So again, this is something that you can't necessarily see, but we are altering it. There are such things as we call microbial pesticides. Our current pesticides, like DDT, insects are becoming resistant to it. I don't know if you know this, but we are constantly fighting against insects for our food supply. And a fair amount of our food supply is lost because insects eat the food, whether it's out in the field and that. You can talk about whether it's good or not and stuff like that, but basically if we get too many insects out there, again, there's been a number of famines that are caused by insects going in and eating all our food, and there's no food left for the human beings. But things like DDT we thought were great to use, but basically we saw that they were affecting not only insects, but they were affecting birds and other organisms. And trouble is DDT has a very long half-life and stayed around in the environment for a really long time. And we banned it in 1972, but people are still finding DDT out in the fields today. Microbial pesticides are ultimate because they are biodegradable. They will totally go away after a bit of time. And they are a great way to control insects. Again, we've identified over 100 microbial pathogens for insects. The great thing about these is they can be genetically altered to increase their potency or increase our ability to control them, so that, again, we could spray the field. It would cause death in that field, but it wouldn't go on to other places. The genes for these toxins that some of these can be placed in the foods. So sometimes these toxins are toxic to bugs, but they're not toxic to us. But we could also do things like, again, the corn hornworm actually eats the roots of corn. And so what we can do is we can have it so that this protein is expressed in the roots and not in the corn. So we're not going to eat it. And so these roots are expressed there. The insects can't eat the roots because, again, they are protected by this particular material, and then we're not going to eat that material. And we see that happening kind of all the time in nature. One such item that I kind of worked on a little bit for a little while is Bacillus thuringiensis. Produces a toxin called Bt toxin. we've actually transformed it into Pseudomonas, and which is an organism that can grow out in the fields very well. And so if you have this gene in Pseudomonas, you can spray it on the fields and it will work instead of Bacillus thuringiensis. And again, working on their way to increase the range of these toxins and to stabilize those toxins. So again, it's kind of one of those things where we don't want the toxins to degrade too fast, but we want them to be around for a while in order to do their work. Viruses and viral pestilence, which we are undergoing right now. Wouldn't it be great if we were able to make a virus that would kill the bugs that are in our field? And we are in the process of doing that. We are looking for that. But again, what we don't want to do is produce a bug that will kill everything else. Kill butterflies that we like, kill bees that pollinate our food and stuff like that. And so we got to look at it. Another problem that people don't like baculovirus a lot is it takes a while to kill the bugs. I mean, you're taking days to kill the bugs rather than you would rather have raid kills bug dead, right? You spray it on the flying insect and it falls dead from the sky. We would rather have those kind of insecticides. Some of the products that we can produce using microorganisms, that we do produce microorganisms, I divide these into what we call primary metabolites and secondary metabolites. Primary metabolites are actually made during the logarithmic phase of growth so that we could do that. Secondary metabolites are made kind of at the end, a lot of times when the microorganisms are stressed. So primary metabolites tend to be things like amino acids. If you take amino acid supplements, we actually ferment and make those. Vitamins, polysaccharides, ethanol, acetone and butanol, which are some of the feedstock chemicals that we use. You don't think about a lot, but in our making plastics and stuff like that, we need lots of acetone and butanol. Some of the secondary metabolites that we actually make, antibiotics, pigments that are used in your papers and books and your clothing and stuff like that. Various toxins, we use toxins to do stuff, alkaloids, many pharmacological compounds are actually made as secondary metabolites, and that is the drugs that we use. Primary metabolites, again, are seen produced during the microbial's growth phase. So if we take a look at our growth right here, and this is looking at ethanol, and so the more cells that we have, the more ethanol that we have, and at a rate they start dying out. And so what we do is, if we do continuous process, you want to take your media right at about this point where you're continuously having ethanol produced. So we'd add a little bit of media, we'd take a little bit of media out, and we would get some ethanol from that. And so rather than just kind of let your big tank go up and get 5% alcohol and then pull it out of there, you're constantly pulling some points off. Secondary metabolites, what we call secondary metabolites, are secondary because they're not required for cell growth or function a lot of times. And so again, when you see primary metabolites, if you stop those, basically the cells are going to die. In secondary metabolites, the cells can actually live for a while. And so what we tend to see is things like penicillin. When we're growing penicillin in exponential growth, we tend to not see the maximum amount of penicillin. We see the maximum amount of penicillin is after growth has stopped for a little bit. Again, primary and secondary metabolites are part of the materials that we can see. We could also see enzymes being produced. So lipases, lactase, protease, amylase, pectinase, TPA. So a lot of your wine, a lot of your juices have pectinase inside of them because the pectinase is kind of a thicker. Some people, you like your apple juice to be really crisp and liquidy, and so you'd put a pectinase in there to reduce the amount of pectin in there that gives it this kind of thick, chunky flavor. Your clothing, in order to keep it washed, you want a lipase, so to get rid of kind of fat stains and stuff like that. We use various enzymes. So a lot of these enzymes are produced commercially. There is a company, so you have Genentech and Genzyme, which is down in South Bay, which is a spin-off of Genentech, was producing some of those. Fuels right now in California, Illinois, where I'm from especially, we tend to see a lot of fuels. A lot of corn is being converted into ethanol, if you can use that in your car. There's a high cost of input, and basically we only get about 12% conversion on those. There's a lot. The idea that we can do is we can use green waste. So again, wouldn't it be great when we go through and we rake out all of our forest and we get all that materials if we can convert it into ethanol? And that would actually pay for that, right? And so here's kind of one of those things where maybe this process can help us relieve something like forest fuels. Because the reason why we go in there, it's expensive to burn the forest fuels, it's expensive to process it. But if we can make something like ethanol from those kind of waste products, then we would have kind of an impetus to kind of go in there Some species like Clostridium and Chlorella actually will produce hydrogen directly, and we could burn hydrogen as a fuel, which is one of those clean fuels, so it's not. The plastics that we produce, we can use living organisms to make the complex polymers of plastics, and so we can make different kinds of plastics that do different activities. All of these would be biodegradable because they are made from biological systems. They can be degraded by biological systems. One of the examples would be polybeta hydroxyl alkylate, PHB, PHB in here, which is basically this structure here. Because it's made by biological systems, we can actually have enzymes that can break this down. This little Pac-Man looking like enzyme, and what would be really great. And so here are some of the things that we can make. PHB is biodegradable, and so you can do something like make surgical implants from it. We put titanium plates in people to kind of hold the bones together, but then you have to go in there and you have to pull out the screws and you have to pull out the titanium plates and people have the surgery. Would it be great if we can make something out of like PHB that would support and hold the bone until it healed itself, and then that PHB would be able to recycle directly from the body? Because it comes from a living system, again, those processes can happen, and so we tend to see this, and I'm not going to really get into the molecular genetics of this. We've been using microorganisms to extract metal for a long time. Actually, Thomas Edison worked on this for a little bit in looking at science. And so metal extraction, extraction of specific metals from flowing water or oceans, the use of specific transport proteins can remove certain chemicals. And so again, ferrooxidants, and we've been using, again, you see these mines where we go in there and we break up these big boulders and we put them in pits and we have these acid pits. Well, we're not making those acids. What we're doing is we use microorganisms that make the acids. So they use the materials and they produce this sulfuric acid, and from that liquid, it's called leaching, we're able to pull out things like copper and iron in order to do that. And so we would normally not be able to use some ore because there's so little. And again, in the past, when we would do our mining, we would look for veins, right, where you actually had your metal in there and you were able to go in there and dig it out and pull out that metal and use the metal. And what we found out is there's present in low amounts in all the rock around that vein that actually had your particular metal that you wanted. And so by using microorganisms to bump up the acid, we're able to release those metals and then harvest those metals again. And so, again, by using very big trucks and all this stuff like that, we're able to get, gram quantities, gram per ton quantity. So you have a ton of ore and you have a gram of material, where in the past you wouldn't really be able to do that. You wouldn't get enough money on it. Because think about it. How much do you spend for a gram or a penny? Right, that copper and that metal that's in that penny, if you had to use a ton, if you had to move a ton of material to make that penny, it'd be way too expensive to make the penny, yet we can do it. Why? Because microorganisms are doing the heavy lifting for us. Thank you, microorganisms. Okay, again, what can microorganisms do? Microorganisms do all things that we currently use chemistry and energy to do. We just don't know how to use the microorganisms yet. We don't know how to ask the microorganisms how to do this yet. And so this is great. And so this is a little bit why they call it practical knowledge, And so in the future, we will use microorganisms to do X. And so one of the things I'm going to ask you guys to do in class is to think of a project of what we can use microorganisms to do. And I want you to kind of present that material. And we'll talk about it a little bit later. But some of the things that you may talk about, some of the things that I will talk about here, is that microorganisms can be used to convert waste into usable items like energy and food. Wouldn't it be great if our sewage treatment plants, you flush, you produce this thing that you put in your toilet from your backside. It's totally waste. It's gross. It's disgusting. Yet, if we can use that thing as a raw material to make things that we want, like steaks or clothing and stuff like that, that would be a good thing. Again, we can use microorganisms to harvest metals from our oceans. Like I told you before, in that there's the largest supply of gold outside of Fort Knox is actually in our oceans. The trouble is, it's present in such small amounts inside of our oceans that it's hard to, it's not worth processing it in order to get the gold out of there. Could we get the gold out of there? Yes, but again, it's very expensive. Again, if we would train microorganisms to pick the gold for us, they would sit there and metabolize and pick the gold for us. We could probably set them out, come back a couple of years later, and grab our gold. Again, microorganisms can clean and remediate toxic waste. We're using them to do that. They can deal with hazardous materials that currently cannot be contained. And so we produce some acids that are so strong that anything that we put them in, they melt out of there. Yet we can use microorganisms that can neutralize these things and can possibly contain these things. Again, microorganisms help us produce our food, our fuel, and our forest for housing. And so I was looking for that, the 4Fs or something like that. And so we human beings, in order to survive, need a number of things like food and fuel and housing. And microorganisms, I think, could be very good at that. So, in summary, and I'm going to go on this a little bit. I have a little bit more summary items to go after this. But we're on the verge of an age of discovery. And this is similar to the one that we went through about 5,000 years ago. And again, the numbers are different. I've seen numbers 12,000 years and stuff like that. But at certain points, we human beings didn't know really how to grow plants. We didn't know how to ranch cows and grow chickens and stuff like that. And so roughly about that time, rather than kind of going around and hunting and gathering things that were there normally in the wild, what we were able to do is like, hey, if I sit down and I grow this grass and I protect the grass and I watch this grass and I protect this grass, I'd have more time to read books, or it wasn't read books, I'd discover the written language. I'd have more time to discover government and kind of stuff like that. And so we tend to see, and so our numbers of human beings kind of went up as we were able to do this. Again, just like we had that revolution that 5,000 years ago, right now we're on the verge of another revolution. Can we use microorganisms to make our lives better, easier, and healthier and longer? And so what are some of the services that microorganisms do for us? And the big one is preserving food. And this is old technology, right? That's looking from the 1800s. I mean, we're looking at food safety. We're looking at food preservation. How can we keep our food longer? We're looking at health maintenance. I'd like to cure disease, but I'd prefer that we didn't get the disease to begin with. Waste processing, that's our sewage treatment. Biomineralization. Again, we need to remove, we have a little bit of excess carbon dioxide in the atmosphere. Wouldn't it be great if we can remove that carbon very cheaply and easily? And that's a possibility with using microorganisms. Bioconversion, there's many things that are out there that we need to make, and we can actually, wouldn't it be great to take a waste product like the grass that we cut at the side of the road and just throw, or we let sit there and rot? Wouldn't it be great to be able to convert that material into something that we can use? Another thing is I'm saying here is that environmental services that we don't recognize. And this day and age, we human beings hopefully are getting a little bit better at recognizing like the environment itself serves a purpose and has an ecological purpose. And a lot of times we don't think about it. And so what we're seeing is that there is a value that's out there, and so the production of oxygen, which we don't control or regulate and stuff like that, which our lives depend on, is out there, and microorganisms are involved in that. And so, again, as we human beings get a little bit better, and definitely as our number grows, we're going to need to look at that. So in preserving our foods, the public health process is preserving our foods. So again, we got a lot healthier when our food got better, when our food got well preserved. We got a lot healthier. Number one, every 5 years we didn't have a famine. A whole bunch of us didn't die of starvation. Even here in the United States, when the whether it's good or bad, when the Pilgrims came over, that first winter, half of them died of starvation because they weren't able to get enough food. They weren't able to bring the food with them from England. And so we have something that's called Hazard Analysis and Critical Control Points, HACCP system. And so basically what we're doing with this is the federal government says, hey, we got to start looking at our food and looking at the hazards that we have from there. And I'll talk a little bit more about this in a second. But basically we're trying to safeguard our food from farm to fork. And one of the big things is, where does our food come from? For a long time, you went to a store and you had no idea where your food came from. And, Molly Stone's and some of these other stores that were a little bit more shishi or whatever would say, oh, this is from Bob Smith boat in Northern California, etc. And, if I had a point, it was, oh, we want to know where our food comes from. But where we want to know where our food comes from is if there's some toxins or something like that, we need to be able to follow it. Or we need to be able to see if somebody gets sick. We need to be able to see who else has had that food so that we can recall that food. And ideally, we don't want to have that happen at all. And so a big thing of what we're looking at is this kind of thing, prevention. How can we prevent things? And so, again, this HACCP is designed to prevent contamination, to identify that prevention when it occurs and identify it before it gets out to the consumer. And so this requires monitoring your temperature, monitoring for microorganisms. And so there are lots of jobs out there for microbiologists to be monitoring this, and we tend to see this a lot. President Obama signed in what we call the Food Safety Modernization Act. This is one, I like it and I don't like it as well. It used to be, we used to make a lot of money by kind of like boiling up hot dogs and selling them on campus. And, we can't do that anymore. Because again, you have to have some way of controlling the microorganisms. People have to be safe. If you work in food safety and you have to take this training, all that is part of that Food Safety Modernization Act. And rightfully, I should say rightfully, I should change my mind saying rightfully. We have roughly about 3,000 people would die each year due to foodborne illness. We have about 128,000 people a year die. When 200,000 people die of COVID, but basically in the long run, this is kind of really big and serious. And 1 out of 6 people get sick a year. I mean, it used to be you get food poisoning. Hopefully, we say that a lot less. I mean, I had one time when I was in graduate school, I had some bad Ethiopian food, and I had to go to the doctors, and I had to go to the hospital and get, and get an IV in because I was losing liquids out of both ends so quickly that I was getting dehydrated, and it was very serious. And so the Food Safety Modernization Act hopefully prevents a lot of that from happening. Again, that's Centers from Disease Control, the CDC, which, again, we've seen in news, which is kind of getting politicized. And so at a certain point, these things weren't politicized. You had these professionals, you kind of going in there and working on it, and we're kind of seeing problems when we have these kind of things be politicized. And some of the problems that we have now are directly due to people who don't know what they're doing or people who are political kind of controlling things. And so, again, people getting sick and people dying from foodborne diseases is largely preventable, and we have the resources to prevent that. And so again, it's not free, it's not expensive, but it's not free, but it costs us a little bit of money. But we got to say is those 3,000 people are important. So the Food Safety Modernization Act calls for the producer of food to actively monitor the contamination of food. And so a lot of laboratories were set up where microbial, chemical, and other items are tracked. The food are prevented real time from kind of reaching the consumers. So you too can get this job, although they're not wearing goggles and stuff like that. So we're this is like looking at juice or something like that, and you're looking for microorganisms in there. And so what we need to do is, in the past, we would kind of we would handle food a certain way. The food would go out. If a bunch of people got sick, we would go back and we would recall that food and stuff like that. But now what we want to do is we want to prevent that from happening, right? And so we're looking for the microorganisms before they go out, and so that material is safe. Again, another thing that we need to do is microorganisms are out there. They're always kind of eating the food, and they want to eat the food that we eat as well. So preserving food is synonymous with preventing growth of microorganisms. We have some old technologies that have been around. They're not really old, but canning is steaming under pressure. We use an organism called Clostridium botulinum to test. It's a test organism. We know if we can kill Clostridium botulinum, that we can kill almost anything that's in there. Some endospores and thermophiles can survive. And then look at the cans. But again, if you look at the grocery shelf, all that food that's in there is all preserved. We have a number of different ways. We have another system that's called aseptic packaging, which I kind of really like. And in aseptic packaging, what you do is you sterilize your material, and then you put it all together, you package it. So you don't necessarily have to use preservatives. Which I'm not saying are a bad thing, but I would like to reduce the amount of preservatives. And so a lot of here's all the juice boxes that you make. Here's somebody inspecting the juice boxes. But basically what we do is we sterilize all this material. And so we have this piece of equipment in there. It's absolutely sterile on the inside. On the outside, you can have people with exposed skin and stuff like that. But on the inside, all these materials, the juice going in there is sterilized, the packaging material is sterilized, it's assembled, and then when it comes out, you basically have this sterile package, and as long as you don't puncture the package, it's okay, and you don't have to do anything else. Another thing that we can do is use gamma radiation. So we can use radiation to kill microbes. So you can package things. Doesn't matter if it's a little bit dirty. You shoot it with radiation and it's there. And in Europe, they do this with milk. And so you can in Europe you can buy milk that's packaged in a bag. It's irradiated and its shelf life is good for months. Whereas we need to keep the milk on frozen. We do pasteurize milk, but milk will definitely go bad. Another problem with radiation is great because it will kill insects, parasitic worms, anything that kind of goes in there, anything that has DNA, it will scramble and it will kill it. So what I want to tell you about is just some of the groups that are out there. We have a group called the Food and Drug Administration, the FDA, that's responsible for developing these processes. And so when you want to sell a food, you have to show the FDA that what you're doing is safe and kind of ready to go. And there's a YouTube watch that we can work on there. The Centers for Disease Control is also involved with this as well, and so they tend to look a lot more at like outbreaks and stuff like that, and how do we prevent some of those outbreaks, right? But and again, the FDA is the CDC is actually moving into areas where they didn't before, and so what we see with COVID, that the CDC is saying no, you can't if somebody can't pay their rent because they lost their job because of the pandemic, you can't necessarily kick them out because that would be a health issue on there. And so it's really kind of interesting of how these things are kind of spreading and going out. In summary, these are some of the processes that we will kind of discuss. I'll explain how microorganisms are used to clean water and oil spills and sewage treatment. We'll discuss these on the discussion board. Describe how organisms are used in the fermentation process to make our foods, beverages, and biofuels, and list examples of microorganisms. So I wanted to go and say, is microorganisms do this? So some of the major characters. So in summary of this talk, what I want to say is microorganisms do stuff for us in our environmental. And so bioremediation. So microorganisms are able to take some of the poisons and toxins that we produce in our industrial processes, but even things like your local gas station that you have, which has this benzene that can kind of leak into our groundwater. They can be microorganisms, if done right, will do it naturally, but for us, we can make them. Sewage treatment is a big one. We tend to think about us extending our lifespan because we develop antibiotics and stuff like that, when actually the reality is we develop better sewage treatment plants. We don't poop where we walk and eat and all that kind of stuff like that, and so that tends to be a little bit more. Crop safety and improvements, we're trying to do this as much as we can. Again, a lot of this happens, we tend to see a lot of the lettuce and stuff like that. There's deer that are kind of running into the field and pooping in the field, and if the deer have listeria or the salmonella and stuff like that, it kind of goes into our food. We're hoping to kind of monitor and look at that. What I'd also like to say is that, in California here, we had a record year. Millions of acres were burned. And again, microorganisms are involved in the recovery of our forests. And that if we pick the right microorganisms and seedlings and everything else to put inside of our forest will come back a lot faster. Will our forest come back without this? Yes, but it takes a lot longer. With a little bit of human intervention, we can have a natural forest come back a little bit later. What I'm showing you here is this is just a process of sewage treatment. And so you don't think about it, but you flush your feces down the toilet. This actually goes to a place, a central place. All the pipes kind of lead to a sewage treatment plant. We don't just dump it out in the ocean. We used to do that, but then we had lots of diseases and stuff like that. And so what we do is we treat it. And so we take that, we grind it all up. We then let it sit for a little while. The solids that come out, we put into what we call a sludge digester. The liquid that's coming off of that is usually kind of brown and has lots of nutrient organics and stuff like that. And it will suck up a lot of the oxygen and stuff like that. And so what we do is we put it in tanks. We shoot air into these tanks, and we let the microorganisms grow in that. The microorganisms grow, and they take out all the material, and that leaves us with a little bit of sludge. We could put that in more sludge, and then when we take this liquid off in here, basically a lot of times we pour a little bit of bleach in it, disinfectant in it, and it's ready to go. And in reality, it's drinkable. We tend to do things with it, and so again, in Burlingame, if you go by the Costco over there by the airport, there's a sewage treatment plant, and with their effluent, they just dump it into the bay. In Pacifica, which I can see it right now, we have the Pacifica Pier. There's a pipe that goes under the Pacifica Pier where we dump our water into there. When I was in Chicago, the Chicago Municipality Waste Department would dump it into the Chicago River, and What's kind of funny is that downstream, St. Louis would get their water to drink from that particular river, or river that it flows into. And so these things can be recycled, and we human beings can kind of help with that recycle. But our main thing about this is we want to remove nutrients from here so that it is not toxic, and we won't see a buildup, and we won't see this process that we call eutrophication of our rivers. We also want to get rid of pathogens that are inside of there so that human beings won't do that, and we're getting a little bit better. And again, this process is practiced up in the International Space Station. And so when you go up there, those individuals they're drinking kind of the same water. What they're drinking today may have been sweat or feces or urine yesterday. So they go through and they do a process where they do it. And they tend to be a little bit more chemical than biological. But they have the money to do that. But if we're going to do this a little bit more in the long run, we got to find biological systems that would actually do this for us. So to move food microbiology, I talked a little bit about aseptic packaging. Fermentation is a big one that we have that increases the nutrients and the taste. And so again, that's improving our food nutrition and our flavor. There's a Swedish restaurant company called Noma that does a lot of stuff. The flavoring. We put soy sauce on a lot of our food, and that's all kind of flavoring comes from microorganisms, and improve food production as well. And so impossible meats, we're seeing kind of a change in food. The hemoglobin substitute that comes from there is actually grown in yeast. So this is helping with a lot of stuff. And so, again, we think about this, we tend to a lot of think of microorganisms. We think about our food and we think about our health, but microorganisms play a big role in industry. And so the industrial process products of microbes. And so things like ethanol are fueled. Conversion microbiology, where you can actually take things like corn and convert it into different things. So we're looking at biofuels. Housing. Wouldn't it be great if we could have microorganisms actually have a mushroom grow and produce a house, and some of the energy that we produce. Actually, there was supposed to be a change in here, and I think I'll talk about it a little bit later. There's what we call a new microbiology, or I'm calling this the new microbiology. And so here we have a human being here, and this is supposed to represent the genetic maps of microorganism. And so the human body, we used to think of us as ourselves, but we are actually an amalgam and an environment. And there's lots of microorganisms that are growing on our skin and growing on our guts. We're finding the importance of probiotics. I saw a little blurb the other day that I need to find and get out to you guys, where babies that were born with C-section, what they actually do is they take a little bit of the mother's fecal matter and mix it up with milk in order to give to those babies so that we can inoculate their digestive system. We're doing sequence analysis. Instead of looking at metabolics, we're actually looking at genomics in order to identify and look at things. If you're interested, for a couple $100, the Gut Microbiology, the American Gut Project, you can actually poo in a container and send it in, and they will tell you what microorganisms are in your gut. And I did this last year, and I'm still waiting for it, but hopefully it will come. Microorganisms, are very scary things, and the COVID is scary. But what's even more scary is there's actually something called the African swine flu, and so there is a virus that's going out that's absolutely lethal to pigs. Okay, a pig gets a kind of a runny nose, a fever, and stuff like that, and in a relatively short amount of time, it's dead. We've been having a hard time controlling this stuff, so it's called African because it came out of Africa. But I'm not going to get into that because a lot of times when we call it African, we're kind of blaming them. But basically, we have this swine fever that's absolutely deadly. We have no treatment. We have no vaccine. And so China lost a lot of their pigs a couple of years ago. Almost all of their pigs they lost due to this particular fever. We're seeing it kind of popping up in Europe, and we don't want to have it happen here in the United States. And what the trouble is we have all these pigs on these farms and stuff like that, but wild boars that are around, if they get this virus, they can carry that virus, and it lasts a long time. We'll see it in the fecal matter. There's ticks that are able to bring it about. And basically the only thing that we could do now is basically when we see these organisms, we basically kill them all. And we'll talk about it a little bit later, but it's called the ring process. And so if you have a farm that has one of these organisms on there, you kill every animal on that farm. And all the farms within a mile distance, you kill every animal on that farm. Okay, and it works, but we can't do that for human beings. And so we'll talk about it, but it is kind of scary. So we need to monitor that's what's out there. Another thing I want to tell you about is microorganisms can actually be sentinel organisms, what we call sentinel species. These are organisms, often animals. We think about the canary in a coal mine that are used to detect risks to humans by providing advanced warning of danger. The terms primarily apply to the context of environmental hazards rather than those from other sources. But again, if we use microorganisms, if we're able to train microorganisms, we're able to use them to actually warn us that things are going wrong in our environment and that we need to look at that. So some of the construction microorganisms, actually, there's concrete that can heal itself. And so microorganisms are responsible for basically a chemical reaction. So you would get cracks in concrete that would reduce the likelihood of concrete. And you see our roads and stuff like that. But these microorganisms, what they can do is they can actually take calcium and carbon dioxide from the atmosphere and they can remake that concrete. So when a concrete cracks, the microorganisms in that crack can kind of heal that concrete. So imagine having concrete that would last essentially forever. So the microorganisms are continuously growing it. Microorganisms can actually mine for us gold and platinum. One of my uncles was involved in this. And so, like I said before, the oceans have the largest amount of gold in them. But this gold is present in, a square acre, you would have one gold molecule. And so wouldn't it be great just to have microorganisms? They're self-replicating, just sitting there and collecting that gold particle, waiting a year, collecting another gold particle. But after several years or several decades, you'd be able to go back there and collect the gold. Now, I'm finishing with this lecture, but I want to tell you about the endless possibilities that we have with microorganisms, technologies that we're just now starting to discover. Microorganisms, again, in the health aspect, we tend to think a lot of microorganisms as being our enemies. No, they're actually our friends. They do a lot of stuff. If we know how to communicate with them, if we know how to interact with them, basically they can improve our lifespan, they can improve our quality of life, a whole bunch of different things. And so again, these microorganisms will help us if we just learn how. And so we need people like you kind of going out and studying them in order to figure out how to do this. We can't just leave it to Yale and the think tanks and the big companies like Genentech. We actually have to start doing it on a small scale. And that's one of the things that I believe. And so thank you very much for listening to me. This is the end of the lecture.